is that we will hopefully be able to test on Thursday. So we're going to look at enzymes. Enzymes are very, very important. They're involved in any of the chemical reactions that are in our bodies. So I have sort of a tab there at the first. Just a minute. So this is a graph of enzymes or of reaction rates. And if you notice, this is going to be energy. So if it's the higher up it starts, the more energy is needed for it to start. And then it's going to reach a maximum point when it's doing its maximum amount of reaction, and then it'll decrease to a little amount. So those are the sort of a graph of your reactions. So chemical reactions, those of physical science, is the breaking and creating of bonds between different substances, and it needs energy. It's going to require some energy to do this. And this energy is activation energy. So it's the amount that's needed to get the reaction to start. And so your activation energy is this, was that blue line there. So the energy right in here is activation energy to get that reaction to start. So reactants, these are called substrates where we're talking about enzymes because it's what the enzymes are going to attach themselves to, and it's what is going to be broken down or built up. And it's the thing that is being changed. It could be one substrate or multiple substrates. Products are the things that are produced. Reactants are always on the left-hand side. Products are always on the right-hand side. So you may have to label a graph like that. So there's two types of reactions. The reaction will absorb energy or it will release energy. So two possibilities. So endothermic, endo means to take in, so that's absorb, therm means heat. So it's going to absorb heat energy. And this is going to be like photosynthesis. We're actually going to talk about this in biology in a future unit. Photosynthesis is an example of an endothermic. It's going to take in, absorb in sunlight, and it's going to store it into bonds of glucose. Exothermic, exo means out, therm is your heat energy, so it's going to release energy. So that means bonds are going to break so that the energy is then released. Cellular respiration. This is what a graph for endothermic looks like. Make sure you label it in your notes. Make sure you see that more energy is in the products than the reactants, so label that part in your notes. If there's a graph there, it may be above the notes. I'm not sure if it's right side by side. It is underneath it. So here's endothermic. Underneath it is the graph right here. So you want to label that there's more energy. Notice how high up here, if you ran a line over to energy, it's got a lot of energy. So products is storing energy. It means something was built, bonds were made. Exothermic is the other column right here. And notice that this line is closer down here to the x-axis. That means it has released energy. So exothermic will look like this. The reactants will contain more energy. This is so sensitive. than the products. Here, the reactants have less energy than the products because the energy is going to be put into the bonds to make that substance. Everybody got those graphs labeled? You good, Alex? You good, Amber? Or almost? Yeah, we'll cut it on for just a little bit. All right, so then we're going to look at the two chemical reactions that we're going to study about in a future unit. This is photosynthesis. You will have to be able to recognize this chemical equation. You don't have to be able to write it out. You just have to recognize that it's photosynthesis. Energy is being stored as a sugar because C6H12O6 is glucose, which is a simple sugar. So you're taking carbon dioxide and water and energy from the sun, and you're combining those to get your glucose sugar with excess oxygen that will be released. So endothermic. This one is cellular respiration. And if you look at the two equations, chemical equations, notice that they are reverse of each other. So this time you're taking the glucose molecule, it's being broken down to release ATP, which is a form of energy our body uses, and it is exothermic because it's releasing energy. We'll have a whole unit on just those two processes. And like I said, you'll have to recognize these two chemical equations. You don't have to write them out. You just need to know when carbon dioxide and water is on the left, it's photosynthesis. When carbon dioxide and water is on the right, it is cellular respiration. All right. Enzymes are a type of proteins. And these are very, very important. So if you got the paper copy, it's the next page. I have a repairman at the house fixing my dryer and refrigerator. All right, so enzymes, you must know they are a protein. If you see the word enzyme, think protein. Very, very important. You need to know that. What they do is they speed up reactions in your body without needing a lot of energy. So they do what we call lowering the amount of energy, the activation energy needed. If our body, we didn't have enzymes, our body would could not have enough energy or make enough energy for these chemical reactions to take place. Therefore, we would not be able to survive. And then another term for them is catalysts, because usually they're used to speed up chemical reactions. What they do is they will bond or attach themselves to a substrate, kind of like a puzzle, to the reactants, and it will either help break that reactant apart, or it will help bind two reactants together to create a new product. The enzymes are not changed, they're not used in any way, modified, they're released, and they can be used again. So enzymes are used over and over again. And if we don't have enough chemical reactions taking place and we have excess of those enzymes, And I'm sure the body then breaks them down and recycles them. Are we good here? All right, so we compare enzymes to a lock and key because enzymes are very specific to the reactant that they will combine with. So a lot of times we'll just call it the lock and key model. And again, it's like a puzzle piece. So the enzyme would be like a key that fits into a certain substrate. It fits in the active site, and it will unlock or do whatever it needs to do for that reactant. Sometimes we diagram it like this. So this may be the enzyme. This is the active site. This is the substrate. You will see this diagram on a test or quiz. You need to know the part, so you may want to label it on your diagram. The full triangle is the substrate. The border line where the two are joined is the active site. And up here it's orange is the enzyme. Can I move on? Here. Okay. All right, so if we are breaking apart, breaking bonds, we're going to have a substrate and an enzyme. They're going to come together at the active site. The activation energy to do this is going to decrease, have been lowered, so that the bonds are breaking. You see a line in between that triangle, splitting that triangle, and you get two products. Again, you may want to label that, kind of like it's labeled here. So the bottom pieces are the enzyme. Notice the enzyme doesn't change. This is the substrate. This is showing bonds breaking. You've got two products left. If we're going to form bonds, you can start off with two substrates. So you have two substrates that a bond is forming in between, so that when you end up, you have one product. Now, enzymes can get deformed. You need to know this term. It's called denaturization. So if an enzyme is denatured, it has lost its shape. So if it was supposed to be shaped like this, it is now shaped this way. It's not going to work. So this is how we lose some biological activity. A lot of the genetic diseases that children are born with sometimes involve enzymes that are deformed, and so some things won't function properly. There's, I think, one of the muscular dystrophy, maybe there's several different muscular dystrophies, and the enzymes, there may be a malfunction in a type of enzyme that is going to lead to your muscles not being able to work properly. Insulin, diabetics who, the insulin, they don't make the insulin enzyme correctly, something is deformed there in that making process, then they can't break down glucose, and then they have a type of diabetes. So enzymes have to be at specific pHs, specific temperatures, specific ion strength and solubility. If any change in this, very extreme, it can cause denaturization. That is why when you have a fever and it gets up to at least 104, you've got to get it brought down, because if your body stays at that temperature or higher, your enzymes will start to denature and your body will stop functioning. So if you're diabetic and your levels get too high or too low, you could go into it's too acidic or too basic. Your enzymes will start to not function and your body will stop working.
My brother has diabetes and like we learned that a lot, that because if he doesn't if his sugars get too low, he can go in the shop or whatever.
Yeah. So it's real important, especially for diabetes, that if you have that you are eating and monitoring your sugar, because you want your body to be able to function. Now, sometimes enzymes can be renatured to their original shape, but not always. Usually it's not reversible. All right, so when we talk about chemical reactions, again, this is a review from physical science. We can increase temperature, which increases the rate of reaction. So when molecules are moving faster, colliding with one another, more often, it can increase the reaction time. pH, how acidic a solution is, or how basic, can increase reaction time. Most enzymes work at a very specific enzyme, so it's very, very important, as well as temperature, that those stay within check for normal. Substrate concentration, how much of the reactant is there? If there is more, you're going to have a faster reaction time. And then last, catalysts, like enzymes, are going to help speed up reactions. So they're going to lower the activation energy, and that causes the reaction to start sooner and get through. Now, sometimes you have a competitive inhibitor. Remember, to inhibit means to prevent or to stop, and this is going to slow down a reaction. So if there's another enzyme that also works on this particular substrate, maybe they're competing, and it may slow down the reaction rate. I think that was it. So hopefully tomorrow we'll have some kind of activity for us to do with enzymes. Wednesday will be kind of a review day. Thursday, we're going to test. And then Friday, it will be a combo of get caught up versus, and maybe a little preview of what the next unit will be. So we have about 10, 12 minutes left before lunch. Work on anything that's missing so we can get caught up. Make sure I get your enzyme notes. And I think that's it. Ian, if you don't have any questions, you are free to sign off. Not sure if you're talking because I'm, for some reason, not picking you up.
Hello.
Oh, there you are. Real. I don't know if I need to turn this up. Maybe I now. There we go. Any questions?
No, ma'am.
Okay. Try to work on some work for me, okay?
All
All right. That recording.